Hello and welcome all in this video. So I am Dr. Jay Kumar and we are going to discuss about the life history or the life cycle of the Ascaris lumbricoids. In that, the first stage which are found, that is the copulation. This copulation which takes place in the small intestine of the man. So man is a host of this parasite. That is, the copulation takes place in small intestine of the host where the adult worms are live. With the help of the pineal city of the male, the pineal city which is found towards the posterior end of the body of the male, with the help of the pineal city of the male, which can open the vulva of the female. In this way, sperms are transferred into the vagina from there. These sperms are passed to the proximal end of the uteri. These sperms then enter into the proximal end of the uteri, and where there the fertilization takes place. The fertilization takes place in the proximal end of the uteri or the distal end of the oviduct in the body of the adult female ascaris. This is the first step in the, and that is in the fertilization. From after the fertilization, the zygote is formed. This zygote or the fertilized eggs moves down the uterus, and around that particular zygote, uterine wall secretes a thick, hard, yellow or brown albuminous coat, and that coat, which is a proteinaceous coat, around the eggs, around the fertile eggs, a proteinous coat is secreted or formed with the help of the uterine wall, which is hard yellow and brown colored albuminous coat is formed around that particular fertile eggs, and which is having wavy surface. This particular coat of the eggs having wavy surface, and this type of eggs elliptical and about 60 to 70 micron in diameter. So uteri of a single mature female may contain 27 million eggs. A single female may contain 27 million eggs with an average daily production of 2 lakh eggs. These eggs, then this type of eggs when formed, these legs then leaves mother's body. These eggs then leave mother's body, release mother's body. That is, they comes out of the body of the female, where they comes then into the intestine of the human being. They leave the mother's body through gonopore and comes into the host intestine, and finally pass out with the host waste material. So these eggs then pass out with the host waste material, comes out of the body with the host waste material. Then these celled eggs remain alive. Such type of eggs, this type of celled eggs remain alive in moist soil for about several years under adverse condition, and under suitable condition of temperature, moisture, oxygen, these eggs undergoes cleavage and develop into infective stage. They can remain alive for under adverse condition for many years, but when the suitable condition is appears, then this undergoes cleavage and develop into infective stage. So afterward, this one celled zygote develop into many celled embryo. So at the time of cleavage, this cleavage is a spiral and determinate type of cleavage is found in this organism. In spiral and determinate type of such type of cleavage, the first division occurs. At the time of first division results into two cells. That is, after in the first division, two cells are formed. That is dorsal cell and ventral cell. Here is a dorsal cell and ventral cell. In the first division, results in dorsal cell and ventral cell. Dorsal cell, which is called as AB cell. Dorsal cell is called as AB cell. Ventral cell is called as P1 cell. So one celled embryo, one celled zygote then develop into dorsal cell AB and ventral cell P1. Again, dorsal cell divides into an anterior A cell. Dorsal cell divided into anterior A cell and posterior B cell. Dorsal cell divided into anterior A cell and posterior B cell. While ventral cell divided into again two cells, that is upper cell which is called as EMST and lower cell which is called as P2. So again this is P2. So again ventral cell divided into upper EMST and lower P2 cell. So at the time of first division, dorsal AB cell is formed, ventral P1 cell is formed, dorsal A1 cell then divided into anterior cell A, posterior cell B, ventral cell P1 divided into upper cell MST and lower cell P2. This four-celled embryo, this now there is having four-cell stage, and this four-cell stage looks like capital T-shaped. This four-celled embryo looks like T-shaped, but soon becomes rhomboidal. T-shaped cell, T-shaped four-cell stage then become rhomboidal because P2 comes to lie posterior to EMST, because this P2 comes to lie posterior side of the EMST. So this become a rhomboidal shaped structure. Again in the next stage, some dividation also occurs in the next stage, that is A cell. Again this A cell is divided into again two cells, that is called as A1 cell, A2 cell. A cell divided into A1 and A2 cell. B cell divided into B1 and B2 cell. B cell divided into B1 and B2 cell. Then EMST cell divided into MST and E cell. EMST cell divided into MST and E cell. MST and E cell. EMST divided into MST and E cell. While then again further E cell divided into E1 and E2. E cell divided into E1 and E2 in the next stage. P3 cell divided into P4 and D cell. P3 cell divided into P4 and D cell, and again P4 cell divided into G1 and G2 cell. So such type of dividation is formed, takes place continuously, and afterward these cells divide, redivide and forming a bunch of cell, and that is called as a gastrula, blastula, and then that convert into the gastrula. So this is a blastula. This blastula is formed after dividation, redividation of the cell. Blastula is formed, and this blastula is 16-celled embryo. 16-celled embryo is called as a blastula. Before that, here is the fate map, about the fate map of various cells. So the fate map is fixed, that is from the A and B cells ectoderm is formed. Ectoderm of the embryo is formed, or the ectoderm of the larva is formed. From the MST cell mesoderm is formed, and ectoderm of the foregut is formed from the MST cell. E1 and E2 cell can give rise to endoderm. G1 and G2 cell give rise to germ cell, primary germ cells. C and D cells give rise to ectoderm and mesoderm. So such type of fate maps are fixed in this particular type of development of the Ascaris. Then again this 16-celled embryo from a hollow ball-like structure, that is called as a blastula, and there is a cavity under that. In that ball-like structure there is a cavity. That cavity is called a blastosole. This blastula undergoes imagination. This blastula, one side of this blastula goes down into the under that ball shaped structure, that is called as, and that forming a structure that is called as gastrula. So blastula undergoes the process of invagination from gastrula. It grows in size and to become active juvenile stage in 10 to 14 days. Active juvenile stage is formed. It is also called as a rhabditiform larva. So this rhabditiform larva is now formed from the gastrula. Gastrula grows in size and forming a larval stage. First larval stage is called as rhabditiform larva, which is a non-infective stage of the larva. This rhabditiform larva again molds to become second stage larva. Then this first stage larva then converted molds into second stage larva, and which is the infective stage of this particular Ascaris lumbricoides. Under suitable condition of moisture and temperature, it can remain viable for about 6 years. Under suitable condition, they can remain for about 6 years in that particular soil, and they now are ready to infect the new host. They can then enter into the body of the new host, where in this new host they can travel, that is egg capsule. Many egg capsules or that type of eggs are found in the soil, and that egg capsule then forming the first stage larva, then come to second stage larva, then this second stage larva forming through the contaminated second stage larva, which is found in that particular soil, remain alive for many days, and the second stage larva, when comes in contact with the food and water material, that food material and water becomes contaminated, and through that contaminated food or water, they then enter into the body of the human being. They can enter into the body of the human being. So when at the time of second stage, at the time of entry in the human body, they are in the second stage larva, infective stage of larva. They can enter through the epithelium, enter in the hepatic portal circulation, and then enter to the hepatic portal vein. They enter into the liver. So in the second stage larva, they enter and in the form of second stage larva. In the form of second stage larva, this type of eggs are, and whenever enter into the human body, then this can live, or they can remove, or they can comes out of that particular hard covering, and they enter into the liver through hepatic portal vein. They enter into the liver, and again it reaches into the heart through postcaval vein. Through the postcaval vein, they reaches into the heart. The postcaval vein reaches into the heart. Then again from the heart, it transported into the lungs. From the heart, they can transport, or they can reach to the lungs through the pulmonary artery. To the pulmonary artery, they can goes into the lungs via pulmonary artery. In lungs, it increases in size, and it then goes into alveoli, and after 6 days, second stage larva molds into fourth stage larva, third stage larva, and fourth stage larva. After 6 days, second stage larva molds into third stage larva, and after 4 days, then results into, or they can convert into fourth stage larva. Then this larva reaches the pharynx, and from where it is coughed up and swallowed again into the gut, and then goes into the intestine. In intestine, it molds for the last time and to become adult. So the fourth stage larva then becomes into the converted into the adult, and then the adult forms, that is young ascaris, and then young ascaris then develop into the adult ascaris, and they forms eggs. And such type of life history is followed by this Ascaris lumbricoides. So this is all about the Ascaris life cycle. So here is a two migrations was found in this organism, first migration and second migration. The first migration and second migration here is found in this organism. So thank you for watching this video.